Hello, everyone, and welcome back to our Inspire seminar series. I'd like to start by acknowledging that as we gather virtually today, most of us at University of Saskatchewan and Canadian Light Source are on Treaty 6 territory in the homeland of the Métis. We pay our respects to the First Nations and Métis ancestors of this place and reaffirm our relationship with one another. So today we have a local speaker, and I'd just like to touch on some housekeeping before I introduce him. So Inspire Seminar Series is a series where we bring in synchrotron-related or synchrotron-affiliated researchers so that they can talk about their research and how the synchrotron is involved, talk about different synchrotron techniques, etc. And so if you have any suggestions for speakers, please let myself or Ardelin or Lenore know, and we will add them to our list. For all of the attendees, please keep your video and mic off during the presentation. This helps to reduce bandwidth and distractions. If you have any tech issues, please message Ardelin or Lenore. And if you have questions for our speaker, you're welcome to put them in the chat or to hold on to them at the end where we have a Q&A session. So at this point, I'd like to introduce Dr. David Cooper. He's a professor in the Department of Anatomy, Physiology, and Pharmacology at the College of Medicine at the University of Saskatchewan. He is also a former Canada Research Chair and current Centennial Enhancement Chair in Synchrotron Sciences at the university, and also one of the beam team leaders of the Biomedical Imaging and Therapy Facility at the Canadian Light Source. During his training, he completed 2 degrees at the University of Saskatchewan, one in paleobiology and the other in anatomy and cell biology, and then earned his PhD in archaeology and medical science at the University of Calgary and pursued his postdoc training at the University of British Columbia within the Center for Hip Health. His interdisciplinary research focuses on high-resolution imaging of cortical bone microarchitecture and related applications in the study of bone adaptation, aging, and disease. Today, he's going to talk to us about some insights into lead exposure from archaeological skeletal remains and some reflections on a decade of synchrotron-based collaborative inquiry. So thank you, David, so much for joining us today, and the floor is yours.
Okay, yeah. I just want to say thanks for the opportunity. I've got this labeled as Redux, so this is a sort of a bit of a do-over from the presentation that I gave in the workshop in the spring. That was a little rushed, and we had a lot of technical difficulties. If anyone remembers or who was there, the internet went down, and so people that were trying to join by Zoom we couldn't record things. So we soldiered on, but it was a little bit rushed and not ideal. So I appreciate the chance to come back. So I put Redux here as the revisiting. So this is the director's cut of that presentation. And Linda's already given a land acknowledgement, but I always include it in the talks, including a map so people know where Treaty 6 actually is, as well as Saskatchewan. But okay, so just a really quick outline of what I'm going to try to accomplish today. And if anything's not clear, anybody has a question, by all means, chime in. I'll tell you a little bit about myself, and Linda already did that a little bit. I'm going to give you a really quick crash course in bone microstructural dynamics so that you can understand how bones change. I realize we got a very diverse crowd, so I want to have a bit of a platform so that you can understand how we're using the synchrotron technology to interpret bone microarchitectural distributions of lead in the future. So I'll hit on that, and then I'll talk about the collaborative archaeological work that we've been doing, including work in the Caribbean, the Arctic, and some more modern sort of experimental work, and talk about future directions. So that's my plan. Linda already touched on this. I did 2 degrees here, paleobiology and anatomy, and then went to Calgary and then on to UBC, and then returned actually as faculty here at the University of Saskatchewan in 2007. And there's a large collection of degrees with some strange titles here, but the overall arching theme has always been focused around bone. And since my time returning in 2007, I've done an extensive amount of work at the Canadian Light Source and BMIT, and working a lot with Dean, and Dean's here too. So I always used to joke that I was fortunate enough to come back as faculty and have Dean as a supervisor. So I kind of feel like it's great to have mentors, and so it's been great working with Dean during all of this time as well. So the 2 of us are the sort of co-beam team leaders for BMIT currently. So, I mean, bone is an interesting thing. It's of interest to all of the Tri-Council funding areas. So from a natural science perspective, Bones are a window on the evolution of vertebrates, everything from fish to our own species, but also materials. Material science is interested in bone and how it has toughening mechanisms and other things. So from natural science and engineering, bone is an interesting subject. Obviously from biomedical science in terms of health, and I'll talk a little bit about that as well. Things like bone loss and fractures and osteoporosis. So it's of interest to things like CIHR. But bones are also tied into the social sciences in that our physical remains within archaeological contexts are sort of both biological entities, but also cultural artifacts. And so the social sciences, particularly bioarchaeology that I'll be talking about today, is of interest to the social sciences. So normally I spend the vast amount of my time talking about the natural science and biomedical side of things. I'm only going to touch on it here to give you a context for understanding how we're applying these things in the social sciences. So I'll return to the social sciences in just a moment. So we spend a lot of our time in our lab thinking about how bones age and change with age. People are probably familiar with the term osteoporosis. I mean, osteo is obviously bone, porosis is referring to pores within bone. And it's a disease characterized by a decline in the quantity and quality of your bone, leading to fractures. And so these are some sections, these are microradiographs. So these are physical sections taken through human thigh bones, femurs, of a 20-year-old and an 87-year-old. And hopefully you can appreciate the thinning and increased porosity in the 87-year-old. And this is a progressive bone loss that occurs in humans. Generally, we peak in bone mass in our 30s, and then we decline. And women are particularly vulnerable to osteoporosis due to a number of factors, including one that post-menopause, there's a big spike in bone loss. So that's unique to women. Women tend to be smaller than men, so they tend to have less bone to begin with, and women tend to live longer than men, so they have a longer period of bone loss. People, if you think about fractures, people often don't necessarily think of them in sort of dire consequences, but osteoporosis afflicts something like 200 million people worldwide and costs billions of dollars in terms of treatment and also lost income, et cetera. So 2.3 million Canadians are estimated to have osteoporosis. That's 1 in 3 women and 1 in 5 men will break a bone due to the disease. So everybody knows 3 women, everybody knows 5 men, everybody knows someone that's going to have osteoporosis. And it's so common that it's actually more fractures are more frequent than heart attack, stroke, and breast cancer combined. Not to minimize the impact of those other diseases, but just to give you a sense of the scale of the problem. And again, people think of fractures and they think of bones healing. It's not a big deal, but later in life, a fracture, particularly a hip fracture, can be the event that leads to the downward slope towards death. So almost a quarter of women and a third of men that suffer a hip fracture later in life will die within the following year. So loss of independence, loss of mobility, and also associated frailty. And so as we aim to live longer and healthier, we really have to look after our skeletal health. Okay, so how is it that we lose bone with age? And this is where I'm going to give the crash course in bone microstructural dynamics, and it will become important later. So there won't be a quiz, but I'll bring these terms up later. So 3 dynamic processes are usually described for bone, and they're growth, modeling, and remodeling. And I'm just going to step through them really quickly. So when we think of growth, if we think of longitudinal growth, how do your bones get longer? Bones actually arise from multiple what are called centers of ossification. Hopefully you can see my mouse here. So this is a diagram from Gray's Anatomy of a human femur. And the blue stuff are actually growth plates, cartilaginous growth plates. So these pieces of the bone are combined by these cartilaginous plates, and swelling and growth of the cartilage in there that ossifies and becomes bone is the mechanism by which we elongate our bones. And we've done some work in this area actually at CLS. So this is Arash Panifar's work this is strontium that's going in. Strontium is an interesting element because it'll actually go into the hydroxyapatite of bone. And so if you look here, this is a rat growth plate here. The blank space is where the cartilage is because it's invisible to X-rays. All the blue stuff here is strontium going into new bone on one side of the growth plate. And so growth there extends the lengths of the bones. I included here, I'm not going to talk much about growth after here because when I get to the archaeological stuff, as you'll see, there'll be a lot of discussion about naval personnel, and most of them are done growing. Closely linked with growth is the process called modeling. And modeling is how bones shape and mold their external surfaces, their shapes. And so part of this is appositional growth. The bones get bigger, and you can think about it in terms of like annual growth rings in trees. But it's much more complicated than that. So I've got a cross section here of tree rings that grow, one per year, one ring. On the right here are what are called fluorochrome labels in bone. You inject the animal with a label that fluoresces under a scope, and you can use different colors. There's different substances, and then you know the timing of the color. And if you look here, you can have very complex drifts. It's not as simple as just adding layers on the surface. You can have resorption on the inner and outer surface. The outer surface in bones known as the periosteum, and the inner surface is known as the endosteum. So it's much more complex than simply adding a ring every year. You literally get drift in tissue space. And so bone forming on the surfaces is called modeling. Now, my lab spends most of its time looking at the final one, which is remodeling. And this is a process of internal turnover. And this is accomplished by cells that both destroy and form bone. So osteoclasts are the cells that resorb bone, and osteoblasts are the cells that refill them. And in 3D here, you're looking at a remodeling space. At the tip here, there are the osteoclastic cells boring a hole, moving this space upwards on the screen. And then as the space contracts below, that's the activity of the osteoblasts filling it in. And so you create these cylindrical holes that fill in with bone, and they leave a little blood vessel pore in the center. And that's known as an osteon. And if you've taken anatomy courses, the old term for it is Haversian system. So that's an osteon, or a Haversian system is something right here. You can see there's a border. I'll show you another image of these later. And there's concentric rings filling it in, and eventually you're left with a little canal in the center. So this renews bone, it removes micro damage, it modulates the density of the bone that tends to increase with time. So it keeps bone slightly tougher by making it less brittle. But as you can imagine, this is a process that begins in utero. So before you're born, this is started, and it'll happen until the day you die. But if you keep boring holes in your bone and you don't put enough bone back, you develop larger and larger pores, or more pores, and that's the root cause of osteoporosis, that imbalance. So just to kind of sum up this bit, here's a shot of human cortical bone. Cells resorb a large cylindrical space, so you'll see resorption spaces. Other cells, the osteoblasts, come in and fill concentric rings, closing that space. So this is a forming osteon. And when complete, you're left with a central canal for a blood vessel, and a bunch of concentric rings of bone. And just a few other structures that are going to come up later in the talk is that between these osteons, the original bone that formed with that modeling process can be left over. You can have isolated islands of it, and that's known as interstitial bone. You can also have fragments of osteons because they get superimposed. So from a forensic perspective, if you have a bone of unknown age, there is a technique where you can look at the microstructure and count up the density of these osteons and use that to predict age. It's not hugely accurate, but over time, your bone structure becomes more and more complicated, more and more superimpositions. And the last thing I'm going to point out, it's a little bit tricky to see here, but there's a bright halo around the edge of this osteon, and that's known as a cement line. It's a hypermineralized line where the process reverses. And just to wrap this up, so this is we do a lot of work in our lab on rabbits. This rabbit's been injected with calcine, which fluoresces, and the animal was injected twice, 2 weeks apart. So I've labeled some modeling, both modeling and remodeling are occurring here. The distance between these two labels is how much bone is formed during those 2 weeks. And so if you look in some places, you can see a gap. And the remodeling is labeling the closing of those osteons inside the bone. So, I mean, I'm not going to talk about this too much today, but one of the things that we spend a lot of time in our lab working on are rabbit models of tracking remodeling in 3D. So what you're looking at here is in orange, the pores within a rabbit's shin bone or tibia that were imaged at BMIT at the synchrotron. And then 2 weeks later in green, when the animal has been sacrificed, we scan them in our desktop and we fuse them together, and we can watch the movement of these spaces and infer the behavior of the cells. And this has helped us establish a novel platform for the study of bone diseases and related treatments. And I won't go through this in great detail, but using things like parathyroid hormone, we could augment the speed of advance. Overectomizing animals, or ovariohysterectomy here is a model of osteoporosis. We find they look fairly similar thus far, but there's more of them. And we're pretty excited that glucocorticoid drugs completely disrupt this process, and it's never really been described before. And so this is a team sport and the focus in the lab. And here's the crew of Xuan, Kim, and Lindsay, and Arash, who's a former member of the lab and now beamline scientist at BEAMIT. So we're definitely recruiting and we're pursuing this. So if you know someone who's interested in imaging in animal models, etc., we need them. And instead of wearing a uniform, we just ask that people wear PPE. Okay, that's where I'm going to stop and change gears. And now I'm going to shift to the social science side of things. So hopefully that's a little bit of a really quick background to help you understand some of the stuff that I'm going to show now. If anyone doesn't understand this slide, we'll have to talk about it later because it means you don't know what Monty Python is and we should really have a chat. Okay, so I'm going to shift gears to the third kind of prong of our research program, which is in the social sciences. And I just really want to drive home the fact that this is an extensively collaborative project, and I'm really speaking here on the behalf of many, many others. And I'll try to introduce many of them to you as I talk about the various studies we've done. But this is a really national team effort. And so I'm not going to run through everybody's names here, but many of these people are going to appear as I continue through the talk. Okay, so as Linda pointed out, and I showed you on the slide, I did a degree in paleobiology. I started at the University of Saskatchewan, actually in mathematics and computer science, and quit that promptly within a year and went to archaeology, and then decided that that wasn't hard enough science, and then settled on paleobiology, which was a fusion of paleontology, archaeology, anthropology. So I figured I showed these before. So these are the embarrassing photos. This is my first summer job cleaning out bison skulls in the basement of the Arts Tower. I spent summers doing excavations in Manitoba and Saskatchewan. So this is Manitoba in the bottom. This is near Outlook, Saskatchewan. It was a great way to spend some summers. And then I went on to grad school in Calgary. This is a shot from 2000, I think, when I joined the PhD program. They did headshots. There were no smartphones. There were no digital cameras. So this was a proper photograph that was posted in the department, and got involved in all kinds of things. This is the Oseberg ship. I got to go to Oslo, Norway. This is a ship that had a Viking burial in it. And I spent some time in Puebla, Mexico as well. So in a past life or several lives ago, I spent quite a bit of time doing anthropology. And this is where I sort of got roped into this whole project on lead and bone. And so the inception of the entire project is that a fellow PhD student in Calgary named Tamara Varney, who was a few years ahead of me, was working in the Caribbean. And so we were in grad school there together. And Tamara, who's currently a professor now at Lakehead University, pictured here, she was doing work on skeletal remains that were being excavated at English Harbor Hospital Cemetery in Antigua in the Caribbean. So got a little arrow here. This is the island of Antigua here. Here's Puerto Rico up here, just to sort of orient you. And there was a cemetery that was eroding. Skeletons were sort of having to be recovered to be saved. And what this was a cemetery that was used between the late 1700s and early 1800s by the British Navy. And one of the most interesting things about this cemetery was that it included personnel from both European ancestry and African ancestry, which is quite rare. So to be clear, the individuals of African ancestry were enslaved members of the Royal Navy. So the Royal Navy, they conscripted people from Britain, but they also kept enslaved people of African descent. And so these individuals had been identified here, and Tamara was very interested in understanding how people's identities impacted things like their social status and health and diet, etc. So that was the main thrust of her thesis work. But I remember her presenting in a departmental seminar that lead poisoning had long been suspected as having a major negative impact on the British Navy in the Caribbean. There were stories about things like dry colic, symptoms like sore stomach, neurological symptoms, things that people had historically linked to lead poisoning. So that stuck in my mind. The root cause of this was generally rum, and I'll talk about rum a little bit. Rum and rations and water catchment often used lead-lined things, both on the islands and on ships, for example. So I went back and hunted through my emails, and as early as 2009, Tamara, I was already here in Saskatoon for a couple of years by that point. And Tamara and I began emailing, talking about, wouldn't it be interesting if the synchrotron could be used to probe the lead in the specimens that you've recovered from the Caribbean. And here's a picture of English Harbor. So if you have to do field work, this seems like a pretty good place to do it, although probably better to do it in the winter, quite hot in the summer. But it's a beautiful, beautiful spot. I've never been, so I'm gonna have to find an excuse, and I might get that opportunity, because when I talk about later is we did secure additional funds to continue this work. But yeah, a beautiful spot. Okay, so lead exposure in the Caribbean, and here we're talking about the colonial era from about the 1600s to the early first third of the 1800s. And this is an article by Whitmers and colleagues, and they talk about, I've just highlighted it here, an 18th century Caribbean epidemic. And so it's kind of entered the lore that perhaps lead poisoning or high lead intake amongst British sailors was really impacting their health. And this is one of the figures from that paper that talks about how lead lining and exposure to lead was ubiquitous through the fermenting process. So of course the British were in the Caribbean growing sugarcane and producing rum. And this paper here already hints at one of the problems, and the reason we were talking about using the synchrotron is that bones may be contaminated by surrounding material. So this leads to a subsequent paper by Whitmer and colleagues where they really talk about problems in determining skeletal lead burden, because the conventional way you would do this is you would take a sample, you'd grind it up, and you would do mass spec analysis on it, and you would get a bulk level of lead, sort of a mean gross overall level of lead. But if that lead could be introduced after burial, the question becomes, can you trust those measurements? And there's a few ways to work around it, but in this paper by Whitmer and colleagues, they basically had to give up. They were looking at African-American burials in the States, and particularly in the young people, the lead levels were so high that they just didn't trust them. So the root of this problem is that lead has a very strong affinity to bone. So lead will actually bind to bone's hydroxyapatite, that's the mineral composition of bone. And what that means is that you could actually use bone meal in soil remediation. So this is an ad from a California company here, Lead Abatement Resource Center, where they'll actually come and they'll put a large amount of fish bone into your soil and rototill it in there to kind of grind it up. And what will happen is the lead present in the soil, the soluble lead, will bind directly to the bone and it'll get locked up. And here's an article by Zhang and colleagues just from this year as well, talking about using oyster shell and bovine bone meal for this purpose. So this is confounding for archaeologists, because if there's lead in the soil, it will bind to the bone after life, and we just don't know whether that is a diagenetic process. So in archaeology, diagenesis is used to refer to changes in bones in the burial environment, or whether it's biogenic, meaning that the individual consumed it during life and it had labeled their bones. So this is just an example. So that was, post-mortem lead will attach to bone. In life, if you're exposed to, if you have an exposure to lead and it's in your bloodstream, it will also have a high affinity to your bone. And I'm just showing you images here. These are monkeys that were injected with lead acetate 6 times. And so this is a modeling effect. This is on the inner surface of the bone. So they labeled all 6 labels. And the last one was only 4 hours after administration. So most of the lead that's in your system will end up in your bone or being excreted. So your bone can really lock up lead. Here's just another example from those monkeys, and this is showing remodeling. So this is one of those osteons with labels of lead. So the real goal that we had was to use X-ray fluorescence imaging to map the precise location of lead in the bone, and to see if we could correlate it with discrete microstructural features going back to the microstructural dynamics I talked about, and could we use that to understand what was going on in archaeological remains? Could we start to pull apart what was biogenic and what was diagenetic or in the burial environment? And so you'll notice some of the usual suspects here in terms of, this is a very early work. This is a bone sample that went from Antigua to SSRL. And if you squint, you can kind of make out a very blurry structure in the bone. And there's an XFI map here. There was a very strong lead signal there, and we broke this up in a small Caribbean-specific archaeology journal. And you can see Tamara as the lead. Trina Swanston was a postdoc working with me, Ian Coulthard of the CLS, and Graham and Ingrid, and Reginald Murphy is a local archaeologist in Antigua. So in principle, it worked. So we were excited. And from there, we explored additional mapping. This is a larger piece of bone that was actually scanned at Vespers. And so we have a light micrograph here, and we could map out calcium, lead, and strontium proves to be quite interesting. So in lead, we have to use the L edges, and the escape energy of the photons isn't particularly great. And so we can get some weird edge artifacts. It's even worse for calcium. But for strontium, the escape energy is quite good, and you get really beautiful images of variation in strontium. So this was a British sailor that has osteons that are hot in strontium and interstitial bone and background that are low in strontium, and same with lead. So this is just one of those English Harbor specimens. We found lead enrichment in certain features, so the surface of the osteonal canals and the cement lines bordering them, and often we would find a whole osteon that was hot in lead. And an osteon takes many months to fill in, so variation between different osteons within the bone suggests variations in the exposures and timing of exposures. And when multiple elements show this kind of covariation, and I'll show you a better example later, it's pretty compelling evidence that this had to have been put in during life. So strongly suggestive of biogenic uptake, so we're quite excited about this. And just a footnote on this collection from the English Harbor Cemetery. This is Karen Giffen, who was one of the grad students directly supervised by Tara at Lakehead. And she compared the bulk levels of lead. So not just XFI here, she actually did the mass spec analysis. There were 23 males. It was British naval personnel, so they're all males. 7 were known of European ancestry, 6 of African, and 10 of unknown. And the unknowns are probably because of incomplete skeletal remains, and often you need the face, you need the cranium to actually make a definitive call in terms of ethnicity. So it might have been missing in those cases. And interestingly enough, they found no difference in lead level between those of European and African ancestry, which was quite interesting. And I don't have time to talk about it too much here, but there are studies from the United States from the colonial period, plantation, early slavery periods in the southern US, where indentured servants and slaves were clearly identifiable, enslaved persons were clearly identifiable by having lower lead than those of European and higher class ancestry. And it's been attributed to access to luxury goods like lead pewter, which has very, luxury drinking from lead pewter glasses, etc. So the indentured servants, the enslaved personnel wouldn't have had access to those luxury goods. So it's interesting here that suggests that the exposure in the British Navy was equal. That doesn't mean that these individuals were treated equal, but it suggests that there wasn't a systemic difference in their exposure to lead. So shifting from the early work was just conventional XFI, and we started exploring confocal XFI, which is confocal imaging as a way to get optical sectioning. So you get data, you can kind of reject out of plane fluorescence, which causes blurring. So if you look at the conventional image, it's nice, but it's blurred. But if you shift to a confocal section, you get a much sharper image. And again, you can see the lead in different osteons and borders. And this is the work that was really led by Sangjukta Chowdhury, who's still here at the University of Saskatchewan, I think in research services. Last time I checked and chatted with her. And she was working directly with Ian Coulthard in terms of advancing the actual quality of the imaging. And the confocal XFI was really quite exciting because you could really make out in a sharper way clearly labeled osteons that have higher lead background. This is that interstitial bone, and older osteons that had lower lead, meaning differential exposure. So you begin to get a sense of the scale of the exposure and how long things would have taken. Osteons accumulate slowly over time. So again, I was mentioning like forensic aging, you can get some temporal sense, and this is going to come up in a little bit when I get to the next project. And Sangjukta took this further, so from polycapillary optics to collimating channel array optics, and I don't have a clue how collimating channel arrays work, so someone will have to explain it to me. But the CCA imaging got very high resolution, 2.5 microns, and very beautiful images of elemental distributions in individual osteons. So it was really quite beautiful. So here's a lead map, and again, you can make out things like the cement lines being highly labeled in leads, but some osteons being higher than others. And if you focus on this bottom one here, you can actually make out the fact that there's a halo of higher lead right around the canal, and then it kind of drops and maybe it picks up again. And I mentioned covariation. So here's strontium, and in strontium, you see that bright ring and then it drops. And so these covariation of elements mean that the probability that this is caused post-mortem is extraordinarily low. So what's going on here? Well, this was a British naval sailor. Perhaps they, the variation in strontium here would reflect diet, not lead exposure, but this person would have been traversing the globe, and their diet might have changed when they're on land and on ship, et cetera. And so we could begin to pick up the pulses of this. I think we're only beginning to explore the potential, but it's really quite exciting to see such a fine-grained level of variation. So we were very excited. We were able to do XFI work at very high resolutions, confocal. We were beginning to get hints of timing of how things were done, and being able to isolate diagenesis from biogenic uptake. And we were showing these presentations at the Canadian Association of Biological Anthropology meetings, where we have lots of friends. And one of them started talking with Trina. Trina's really responsible for pulling this whole team together. This is a number of researchers from across Canada. And I'm going to talk quite a bit about the Franklin expedition now. And if you're not familiar with that, I'll give you a bit of a background. But the Franklin expedition sits large in sort of British historical minds and the history of Canada, in that it attempted to circumnavigate the Northwest Passage to make it from Britain over to Japan and China without having to go around either Africa or South America. And so ultimately that expedition was lost, and it's been one of these mysteries that that's been in the press and in the common conscious. So it was really quite a remarkable thing for Trina to be able to pull all of this together, and we received remains from Franklin expedition. I'll talk about that now just to give you a little bit more context. Okay, so what's the Northwest Passage? Just to orient you a little bit, this is Greenland up here. This is Baffin Island. Saskatchewan's right down here. Over here is Alaska. So the Northwest Passage is an attempt to pass through the Arctic Ocean between the Atlantic and the Pacific. And it's only reasonably possible to do this, obviously, in the summer when the sea ice is lower, although with global warming, things are shifting. And this was what Franklin and his crew set out to do in the mid-1800s. So they were tasked with being the first to do this by the British Navy. And I just want to orient you. So Baffin Island here has this thing that looks kind of like a rhino horn. So just keep that in mind if you get lost anywhere. We'll be talking, I'm just going to zoom in where this box is and show you a map of the area. Okay, so here we are. The Franklin and his crew of 134 men set out on May 1845. So mid-1800s. 5 of these men disembarked from Greenland. So Greenland is off your map here, top right. So they entered into Lancaster Sound from Greenland with 129 men. The names of the ship were HMS Erebus and HMS Terror. So at this time, Queen Victoria was on the throne. So it's Her Majesty's ship Erebus and Her Majesty's ship Terror. And I was joking about this last time I gave this presentation. Erebus is like the god of darkness, the son of chaos in Greek mythology. And so I don't know if I'd want to, well, these were British naval ships. These names were meant to strike fear in the hearts of enemies, right? So, but sailing out on Erebus and Terror into the Canadian Arctic maybe was a bad omen. These ships were stocked with 3 years of supplies, and tinning of food using lead soldered tin cans was a new technology, and they brought a huge stockpile of food to be able to do this expedition. So they entered Lancaster Sound, and in the first winter, they stayed on Beechey Island, which is a tiny little island off of Devon Island. So keep this in mind. These 2 sites are going to become important in a moment. So they stopped for the first winter at Beechey Island, and in the next year, they headed down, and they actually got trapped in the ice in September the next fall, 1846. The ships were trapped in the ice, and they were stuck there for 2 years off King William's Island. How do we know this? We'll get to that in a moment. How do you get stuck in ice for 2 years? I'm just going to show you a map. And again, here's Baffin Island. Here's the little rhino horn here. This map you're looking at right now is August 9th, 2013. So August, the sea ice will be at its lowest in September, and then it will freeze back up. So this is kind of getting into late summer. And if you look at the path they would have had to have taken, So this is 2013. You can imagine they would have encountered ice right away here. They never could have sailed down here, and there would have been no clear passage through, even in August. And here's 2016. So global warming's had a bit of an impact, and you can imagine you could maybe plausibly sail through here, get down to King William's Island, and perhaps scoot through. But if you had cold years, the ice might not break up. So they were literally stuck for 2 years in the ice here. And they did expect to be frozen up. The ships were reinforced. They expected they were going to be trapped in ice potentially in the winter. Now, an interesting footnote to all this, we began this project, and at the time we started working on it, the ships were actually found. So this was part of the great mystery of the Franklin expedition. The Erebus was found in 2014, and the Terror was found in 2016. This is Parks Canada surveys. They were using underwater sonar. And so they were reported abandoned, the ships up here in 1848, and they were found some distance away. And so the ships may have drifted in the ice. They had not sunk when the sailors abandoned them, so they were almost certainly moved. And that's what complicated finding them. There's been a lot of interest in finding the ships for a long time. Okay, so I'm just going to dive into a little more detail. So Beechey Island, this is where they spent the first winter. There's 4 graves marked here. 3 of them were from the Franklin expedition, and 1 is from a sailor who died on an expedition that came to relieve them or find them. There was something like 25 major expeditions, either by sea or over land, to find the Franklin expedition or evidence of it in subsequent years. So they overwintered at Beechey Island in 1845-46. There are 3 graves there from the Franklin expedition. They were marked graves, and the men are known because of the markings. And so their names are here: John Torrington, John Hartnell, and William Brain. And in fact, the book written by Owen Beatty, I'll talk about him in a second, and John Giger, called Frozen in Time, has a picture of John Torrington's face on it. They were buried below the permafrost level, so the bodies were essentially frozen and perfectly preserved. So in 1984 and 86, Owen Beatty led an expedition to exhume the bodies and perform autopsies, and they collected samples. The causes of death of these men were linked to tuberculosis and pneumonia, but they did detect very high levels of lead in their soft tissue, particularly their hair of John Torrington. And they linked this to lead solder in the tin food, and others subsequently have said the water catchment or the water storage on the ship. But the hypothesis that lead poisoning led to the loss of the Franklin expedition took hold from these publications. So King William Island, this is that southern site I talked about. This is where the ships were trapped in ice for 2 years, from 46 to 48, 46, 47, 47, 48. A subsequent expedition in 1859 found a note in a cairn off on one of the points, Victoria Point, in King William Island. And it has 2 parts written in it, so I'm not going to go through this all, but it has like a standard part and then the stuff scribbled around the edges. So the first part noted that they wintered at Beechey's and also noted that they were wintering in the ice. This is where it was recorded that they were trapped in the ice at King William Island. And if you look at the end of this little bit here, I don't know if you can see this, it says all well, and it lists Sir John Franklin in command. Part 2 is the stuff scribbled around the sides, and it comes from a year later, April 1848. And it notes that the ships had just been deserted, that only 105 men remained, 24 people had died, including Franklin, and he died in June 1947, which is really only a month after the last entry on the note. And they made for the south, they headed south for a river called Back's Fish River. Ultimately, all of the men died. And in fact, Dr. Owen Beatty had led surveys in King William Island in 81 and 82, well before they excavated in Beechey Island. So we got access to a number of remains from Beechey Island and from King William Island, and we also had comparative material from the Caribbean, from just a few decades before this. And so we set out to answer these questions, or at least test these hypotheses, that the sailors that lived longer, those on King William Island and Beechey Island, would exhibit more extensive lead, that those bone structures forming near the time of death would have higher lead, and that the Franklin expedition specimens would have higher and more sustained evidence of lead uptake than the Caribbean sailors. So I'll step through this really quick. The top two are BI is Beechey Island and KWI is King William Island. So on the top are bone samples from John Torrington and John Hartnell, and unknown individuals from King William's Island. And really the take-home message here was that already early in the expedition, only months into it, these individuals showed very high levels of lead already. That suggests that this lead they'd been exposed to it extensively over their lifetimes, not just in the months acutely after joining the expedition. When we come to the second question, elevated lead in recently formed bone, there was examples of this, and so I'm showing you a couple of them. This is a modeling event, periosteal bone formation, and there's high lead all along this new band of bone. Part of this could be diagenetic, but it does correspond quite nicely with newly forming bone on the outer periosteal surface. And not a lot of the individuals showed signs of starvation or high levels of bone remodeling, but this one did. There's lots of active forming osteons that aren't completely filled in yet, and many of them contain lead. So there's no doubt that there was lead exposure during the expedition, but not all individuals showed this, and it wasn't really marked across all of them. And then finally, when we compare the Franklin expedition specimens against Antigua, both show extensive, like this is King William Island, all of the bone structures in this individual are labeled in lead. This person must have been exposed to lead for many, many years prior to the expedition, as well as during the expedition. So when we looked at the sum of the evidence, and then if you just, this is not the best image, but this is the actual bulk lead. This is done by mass spec. When we look at the Beechey Island, the early expedition guys had the highest level measures, or amongst the highest levels. So that's kind of countered expectation. And in fact, when we measured the stuff from the Caribbean, our highest measurement came from one of the Caribbean specimens. And so taken in the sort of sum of all of this evidence is that the data didn't support the hypothesis that those sailors that died early on had higher levels of lead. It was really inconclusive or inconsistent that bone forming around the time of death was hyper labeled in lead. And when we compared with the Caribbean, the Franklin expedition sailors didn't look particularly high in lead. So the conclusion, or one of the conclusions in this paper, is that in this larger context, the Franklin expedition samples don't appear to have unusually high levels for the historical time period. So yeah, there's a lot of trepidation with publishing such a thing, and it doesn't mean that lead didn't have an impact, but it does suggest that they weren't uniquely poisoned with lead for the time period. But I did mention earlier that in the Caribbean, there were concerns that lead was impacting health. So we have to kind of keep that as a grain of salt for the interpretation. I just want to mention really quickly another Canadian light source connection and give a shout out to Joyce Macbeth, who was on this paper with Jenny Christensen. They got one of the thumbnails from John Hartnell. I'll have to hear the story from Joyce on how they got it. And they did XFI along the length of it, and thumbnails grow, or fingernails grow at a certain rate. So you can look at, if you look here, they've mapped out the tip of the thumbnail, the oldest nail, the oldest keratin was when they left England, and then the newest forming keratin was the time of death. And interestingly enough, if you plot this out, so here's leaving England and leaving Greenland, the level of lead exposure actually drops, or at least the lead in the fingernail drops over time, and then only spikes up again right near the time of death. And there's some reasons for that maybe, but interestingly enough, we don't see an acute climb in the lead levels in this fingernail, which actually fits well with the bone data that we have. So why did the Franklin expedition fail? It's been linked to lead poisoning, as I've said. We can't rule it out, but it doesn't look particularly dramatic compared to other populations. Doesn't fit the timing hypotheses we laid out. The paper that I just mentioned that Joyce was on identified zinc deficiency, potentially linked with tuberculosis and infectious disease. Some people have said botulism, and that they think the tins had incomplete or insufficient soldering, and that the food might have gone bad and potentially poisoned them. Keeping in mind that they were alive and at the ships for 3 years, and their food supply was supposed to be something like 3 years. But if some of it went bad, they might have faced some significant pressure. Scurvy, they took a whole bunch of lime juice with them. This is post-discovery of using lime or citrus as an anti-scorbutic in the Navy. So this is why British sailors are called limeys. They brought it with them, but perhaps they didn't have enough and it spoiled. And there are other possible hypotheses. Tuberculosis certainly being in the close confines of the ship, infectious disease, and the permafrost bodies showed tuberculous lesions in the spines of at least one individual, really blackened lungs. There's lots of evidence of potential causes of pneumonia in those individuals. And then in this paper Byard was talking about suicide and cannibalism, but the conclusion is probably the expedition was lost for multifactorial reasons. All of these things kind of combining, and also being the fact that the Arctic is such a challenging environment. I just want to revisit this cannibalism line first, and that there is evidence of cut marks extensively on a number of the remains that have been found linked to the Franklin expedition. And there were Inuit accounts of cannibalizing of the dead for Franklin's crew. Now, this is Victorian England. They did not appreciate indigenous populations saying that, what was it, quote unquote, the flower of the British Navy would resort to such things. And in fact, Charles Dickens maligned Inuit accounts and had some rather unkind descriptions of Inuit people that even suggested that British naval sailors might eat each other. But there's quite a bit of evidence supporting this, including late stage cannibalism, where extraction of marrow and fat from bones was being explored. So it seems pretty clear these individuals were starving based on the physical evidence and also on the accounts of the Inuit at the time. There were individuals that even suggested that the Inuit had killed them, and there was anger that the Inuit didn't save them, but it's pretty hard to save a crew of 129 sailors when the Arctic is an extremely challenging place. So why did the Franklin expedition fail? Probably all kinds of reasons, but I usually converge on the fact that I don't think you need a fantastic story to explain why people died after 3 years in the Arctic. And in fact, I would argue you need the opposite. It's really remarkable that they could sustain themselves for that long in such a challenging environment. And if anything did go wrong, if their food stores were failing, if infectious disease had hit, other things that would have stressed them even further, it's remarkable that they survived as long as they did. And just as a footnote to this, the Northwest Passage wasn't completed by ship until 1903 to 1906 voyage by Roald Amundsen, who was a Norwegian. And he died in 1928 in a plane crash in the Arctic and was never found. So the Arctic is, again, a very challenging environment. I'm just going to wrap up in the last couple of minutes here talking about some of the next steps. This is work by Rachel Simpson, who's now a PhD student at the University of Alberta, but she was here with us in Saskatchewan, and she took modern cadaveric bone and had exposed it to lead acetate to contaminate it to see if we could replicate things we'd seen. And so here's her modern bone. Here's just taken it and XFI, and then she soaked it in these lead solutions and produced this sort of surface enrichment, and which matched really nicely with some of the archaeological stuff that we had seen. And it didn't mimic or change the overall pattern, but it was superimposed on that pattern, which added some credence to our interpretations. Really interesting from Rachel's work, though, is that these are modern individuals from our cadaveric dissection collection. These are individuals that donated their bodies through the body bequeathal program. And if you look, there's a reversal of some of the stuff we saw in the earlier archaeological bones. Some of the new osteons that are superimposed on the background look really dark. And that's because their level of lead is much reduced. And the interstitial bone is brighter. So the older bone here is brighter, where the newer bone is darker. And we're not sure the root cause of this. I mean, obviously it's declining lead exposure, and it might be associated with the fact that leaded gasoline was banned in the 1990s. And so we might actually see a signal in the bone of these modern individuals for declining lead exposure. And the last little thing I wanted to touch on, I mentioned, sort of alluded to this earlier, we were awarded a new SSHRC Insight grant through the Interdisciplinary Committee, and we're going to expand this work in the Caribbean to look at more of a civilian population and more enslaved laborers as well. And we want to shift to more of a focus on quantification. You've seen most of this is qualitative interpretation that I've given. We haven't talked much about amounts. And we've added a new team member. So Cassidy Vandershee has joined us from the King's University. And we were aiming to use BioXAS all along. And we really want to proceed with focusing on the use of BioXAS rather than APS. And Rachel gave me permission to show these images. Rachel's PhD project now is extending this work to ancient Greece. And so these are bone samples, one's from Sniffilos and one's from Corinth. And these were imaged, and many thanks to Gosha at BioXCS. These were imaged this last summer or early fall, I can't remember. But these are the first glimpses of lead in ancient Greek bones for Rachel's PhD thesis. So I've hit on a number of these people's names. There's too many people here to mention. I do want to add an extra comment in that the individual in the top right is Anne Keenlyside, who was a professor at Trent. And unfortunately, we lost her last fall to breast cancer. So she died just over a year ago now. So that was a real blow to the team, and our hearts and thoughts are with her family and loved ones. To sum up, what started as an unsuccessful SSHRC grant that began with the president's bridge grant at USask has led to multiple Tri-Council grants, students and fellows, helped support research chairs, used 3 synchrotrons, produced 4 theses and 11 papers. And I have 28 presentations here, but it's actually many more now. I guess at least 29 after today. And it attracted a lot of attention. So unfortunately, Trina wasn't around, so I was on Radio Canada. Of course, I didn't speak French, so they dubbed me. I don't know how I feel about being on Fox News, but they featured the story on Fox News. And the images that we generated were actually featured on an episode of Mysteries of the Deep, if you've ever seen that show. So I'm going to leave it there. I think I'm a couple minutes over, so I'll stop so we've got a few moments for questions. Anyways, thank you for your attention.
Thank you so much, David. That was super interesting. I really like to see how you can use different synchrotron techniques to get a better understanding of our like various historical events and how they might have happened. So I found that really cool. So if anyone has any questions, feel free to use the raise hand icon in the reactions, or to drop your question in the chat and I can read it out to David. Don't be shy.
It's been quite the journey. I have the book here. So my wife bought this book when she was an undergrad in an archaeology class, and this is the Frozen in Time book. So I never would have thought that we would be looking at Franklin expedition remains, but here we are.
Man, that's so cool. Dean, I see you have your hand up.
Yeah, that was really great. I thought that was really, really interesting. I was wondering whether or not you could identify the source of lead by some kind of isotopic analysis. Has that been done? I assume there must be lots of isotopes of lead, just given how heavy it is, but just a thought.
No, it's a really good thought. And in fact, Owen Beatty had done some work on this where they sampled the lead solder on the tins and had very similar isotopic signatures in the bone. Now, whether they could, I don't know how many sources of lead there are in England, and I don't know how far they chased that. And the whole debate's gone back and forth on whether the lead solder in the tins was soluble enough or poisoning them. But there has been some thought in that. And there were literally, I think, 8,000 tins or something. So there are lots of samples of that lead. It's a really good thought, because I think there's been a lot of interest in trying to pin down that source of lead, and then whether or not the lead was the root cause of some strange behavior in the sailors and ultimately led to their deaths.
Very cool. Very good.
Any other questions? So there's a question from Yasemin. Is there a way to definitely rule out that lead was the main cause of death?
Yeah, it's a good question. I don't think it can be ruled out. But in the original interpretation, there's always this desire to calculate lead level from bone. And the trick is, it really depends on how you sample the bone, because one individual, if you sample a little piece of it, could be really high or really low. So short answer, I don't think we can rule out an impact of lead on the health of these individuals. Saying that lead killed them, I think, is an overstretch of the data. I think people have converged on it might have been a factor that contributed to their poor health. But identifying an acute, one of the issues is that archaeologically, like a single large acute lead exposure that kills someone would be very hard to detect, whereas chronic lead exposure is the stuff that we see. So we absolutely can't rule it out, but where would such a potent level of lead come from? So it's unlikely, but not impossible.
There's a question from Lenore. Does lead impact the bone health and regrowth?
Yeah, I don't know how much lead you'd have to have to impact the actual bone growth, but it is well recognized that one of the most insidious things about lead exposure is if most of it gets locked in your bones, you can repoison yourself, or at least re-expose yourself as your bones remodel. And in fact, if that fingernail from the paper with Joyce and colleagues, they see this dropping level of lead, and then near the time of death, it goes up. And they've actually argued that as the body of someone who's really sick starts to consume their own tissues, including bone, that they actually got re-exposed from the bone. So that was kind of an interesting argument. So yeah, it's one of the problems is that you literally can re-expose yourself because it's locked in there, and then the osteoclasts come around and resorb that packet of bone, and it's re-released to your blood.
That's kind of scary, actually.
Yeah, it makes remediating it really difficult.
All right, last chance to ask any questions. We'll give people a moment to either raise their hand or drop something into the chat. Okay, well, thank you again. That was an excellent talk, and it was really, really interesting for me. And yeah, hopefully we'll be able to have some more speakers on soon. Just some quick announcements before I release everyone. So we do have a workshop on December 7th, an Inspire workshop. So hope to see you all there. If you are submitting a poster, please add a poster title. Please add your poster titles to the spreadsheet that Lenore linked in her email. And make sure to also submit those posters for printing by December 4th. So both adding the title to the spreadsheet and for any posters that need printing, make sure you have that done by December 4th so we can get everything printed up in time for the workshop itself. And if you haven't RSVP'd, please do so. We want to see you there. Otherwise, thank you so much for everyone who was able to come out and see the seminar. Thank you again to David for presenting, and see you all at the next seminar. Have a good rate.